The overall goal of this video is to show electrophysiologists how to use the program SpikeSorter to detect and sort spikes in extracellular recordings from neural tissue made with microelectrode arrays.
Our goal was to provide a simple solution to this task of spike sorting using a program with a user-friendly interface. SpikeSorter can read files in a variety of formats and offers a variety of pre-processing and event detection strategies in addition to sorting and data export options.
The implications of this technique extend towards the analysis of neural behaviors studied by extracellular recordings with multi-electrode arrays. Generally, individuals new to this method will struggle because of the complexity of the task and the lack of standard methodology and objective validation. We first had the idea for this method when we were faced with the problem of sorting our own recordings done with 54-channel multi-electrode arrays and found no available solution.
To begin this procedure, open the program. Go to File and Open, and then select the recording file format from the drop-down list at the bottom right of the resulting Open File dialog. Next, select the file and click Open. Click OK when reading has finished. The recording signals are now shown in the voltage waveform display. If the recording is unfiltered and contains the local field potential, remove it by clicking on the filter icon on the toolbar. Select High-Pass Butterworth filter, a suitable cutoff frequency, and the number of poles, then press Do It. Press OK, then Done, then inspect the filtered waveform in the display. Voltage signals can be inspected by holding down the left mouse button and then dragging. Use the scroll wheel to zoom in on particular features and out again. Hover the mouse over specific waveform.